parts in chapter 16 and 17 that students find difficult. I already did chapter 17, the natural selection part, but I left the variation part because I feel like it goes hand to hand with the gene control part in chapter 16. And in the end, I might do the part about meiosis and a crossing question or so if people have a problem with this, but I'll be mainly focusing on this part. I always like to start with an overview. So the overview of this part is, firstly, we're going to take how the genes determine the phenotypes. Okay, so we're going to take in the variation that there are different ways a phenotype is affected, either genetic or environmental ways. So in this part, we're taking how the genes can affect the phenotype. Okay, so we're going to take basically 4 genes, okay, that code for 4 different proteins. Okay, and see if mutation happened in those genes, how will this affect the phenotype in like disease causing. Okay, and we're also going to take the part about gibberellin. I collected all the parts about gibberellin in the curriculum in the chapter about coordination in the end. I said like coordination in plants, and I included all the parts about gibberellin, so we can watch it there. Okay, then we're going to take how those genes are controlled. So as I said here, we have 3 genes. Okay, those 3 genes, we're going to take the relationship between the gene and the protein. So of course, if a gene is coded for, it codes for a protein. Okay, so first gene we're going to take is the TYR gene. This gene normally, so firstly we're going to take what normally happens and then what in mutations. So normally, this gene codes for tyrosinase enzyme. Okay, this is a protein. What's the function of the tyrosinase? In the melanocytes, those are the cells that give us pigment. Okay, this is what makes our color darker. So normally, in the melanocytes, this reaction happens. So we're picturing this in a cell. Okay, there is something called a tyrosine. This is an amino acid. Okay, and using the tyrosinase enzyme that's coded by this gene, okay, this tyrosine is transformed into DOPA and then into dopaquinine, and then it will give us melanin. And as I said, this is the melanin that gives us our pigment. So we should know the normal chain reaction that happens. So what if someone has a mutation here? What if there is a random spontaneous change in sequence of genes in alleles here? Okay, this will lead us to a functionless tyrosinase. Okay, so this will give us a tyrosinase that has a different active site and does not work. So this means everything here will be like not happening. So the tyrosine will not turn into DOPA, not into dopaquinine, so we will have no melanin. So how will this look like? How will a person that has no melanin look like? This person will have a disease called albinism. An albinism or an albino person is a person who has no pigment in their body and they look white. Not just like white as a person, but just like no pigment in the hair or in the eye or in the skin, like everywhere. Okay, so this person firstly will have no melanin pigment. Okay, he will have no pigment in the eye. So this will leave the eye kind of in a pink purple color because since there is no color in the iris, okay, you should be specific here, like say a pink eye because there's no color in the iris. Okay, so the iris will show the blood capillaries below it, which is like around pink. The hair and skin are also pale, and the eyes have a jerky movement. Like you can see a person having albino, you'll always see the eyes kind of move a little bit. That's because of the lack of melanin too. The thing you should notice about all of those 3 first diseases is that they're all recessive mutations, okay? So the normal gene that codes for the tyrosinase is a dominant gene, okay? Allele. So if there is a mutation that happens, this new allele is a recessive allele, okay? So for a person to have the albinism, he has to have 2 recessive alleles, okay? Same with the sickle cell anemia and same with hemophilia, okay? So you should notice in your answer that you always write recessive, okay? Because this is a recessive mutation. Then the second disease, we have the sickle cell anemia. Again, we should know what the normal gene is. The normal gene is the HBB gene, okay, that codes for normal hemoglobin. Okay, so here you have to know a little bit more. So the normal base for this, like in one of the codons in this chain is CTT. This is the normal base. This normal base codes for the glutamine amino acid. Okay, so this glutamine is polar, okay, so makes the hemoglobin soluble so it does not clump. Okay, but if there is a mutation and this T is randomly changed into an A, so this will be a cat. Okay, instead of having a glutamine, we're going to have valine. You should know the mutations and know the name of the amino acids. This will have marks. So the valine, instead of being polar, like the glucosamine, no, it is non-polar. So instead, it's less soluble and will crystallize. So if you look to an RBC, instead of being like circular and can pass through the capillaries, no, it becomes this sickle cell shaped. It's like a sickle. Okay, so what is the problem with the sickle cell? Firstly, it has two problems. That it's a smaller, okay, has less surface area. So this means that we'll have less oxygen diffusing, and this person will have severe anemia. Okay, that can be fatal. And then the second problem is because of this shape, it like kind of gets clumped together, okay? So blocks capillaries. Okay, so this can like prevent normal blood from reaching the body and may form clots. So this is a problem in itself. Number 3 is a gene called the F8 gene, okay? That codes for factor 8. Okay, you should write the factor as in like full name here, okay, in your answer. So you write factor 8 and you write the 8 in numeric like this, Roman. And this factor 8 is used for clotting. Okay, so this will help your blood clot. Okay, so if a person does not have the factor 8, okay, they will have a disease called hemophilia. Hemophilia is a blood clotting disease. Those people cannot clot the blood. So those people will have excessive bleeding. Okay, so with any minor injury, those people cannot clot, so they will have excessive bleeding. And this is treated with, in genetic engineering, you took this disease, this is treated by giving them the factor protein. Like you can synthesize this by genetic engineering, like insulin, and give it to them. Okay, so this is a treatable disease, but of course it has to be like re-injected. So what also you should know about this disease, that it's a sex-linked disease. Okay, so males are more likely to get the disease because they only need one allele to get the disease because it's only carried on the X chromosome. Okay, so in this case, they will have a non-functioning factor 8 and the blood will not clot. Okay, so as I said, notice that all those 3 diseases are recessive mutations. And you should write this in your answer. So they are recessive alleles. What about this last one? This last one is different because this one is a dominant mutation. So a dominant mutation means that they only need one allele of this to get a disease. So whether they are heterozygous or homozygous, dominant, they get the disease. Okay, so what is this Huntington disease? So this disease is caused by a problem in the HTT gene. Okay, this codes for Huntington protein. Take care of this because the protein is called Huntington with a TIN, while the disease is called Huntington's with a TON. Okay, so this is a disease and this is the protein. So take care of this. So normally, the allele is on chromosome 4. Okay, I got you this from the mark scheme, so you can like memorize this as it is. Okay, so normally the allele is on chromosome 4. Normally, this allele has from 10 to 35 repeats of CAG. So on this allele, you have like CAG, CAG, CAG repeated from 10 to 35 times. Okay, so this is in a normal recessive allele. If you have the mutant dominant allele, this allele will have more than 35 repeats. So we'll have so many more CAGs. So we'll have CAGs more than 35 times. Okay, the more the number of like the repeats, the earlier the onset of the disease, because normally the disease should come in middle-aged men. Okay, like started like late 30s, like late 40s, like early 50s. But if someone has a lot of like repeats, very numerous repeats, the disease can come in a very early age, like even on birth. Okay, so what happens because of those many repeats of CAG? It codes for the Huntington protein like a lot. So this causes a misfolding of the protein. Okay, what are the symptoms of the disease? They're like mostly neurological problems. They'll have neurological conditions, brain problems. They're gonna have cognitive mood changes and like mental changes, and they're gonna have the motor control is uninhibited, so they have like involuntary movements. They keep accidentally moving a lot, and they'll see them like raising their hands accidentally or moving their leg accidentally. Okay, so this is Huntington disease. please do memorize those diseases because they do not come that often, but when they come, they can come like with 7 to 8 marks, so it's a shame to like lose marks in those easy questions. Okay, I know people tend to forget them because they don't come a lot, but like they do come sometimes. So now that we're done with this, we need to know how are our genes controlled. Okay, first things we need to know what types of genes do we have. We have 2 types of genes. We have structural genes and regulatory genes. So structural genes, from the name, are genes that code for a protein. Those are the exons, okay? Those are the genes that code for proteins or enzymes that are needed for some sort of structural function of the cell, okay? An example of this is the lac Z and Y. Okay, just take care not to confuse the gene with the protein itself. Like the structural gene is the gene that codes. It's not the protein itself. So this is something that I see some students confuse. Okay, and the other type is a regulatory gene. Okay, and this gene codes for a protein that controls and regulates something. Okay, so this protein is not directly structural, like it's not a channel, it's not an enzyme. It regulates something else. It regulates another gene. Okay, so for example, the most common example is transcription factors. Okay, so those transcription factors bind on the promoter. We're gonna take this part in details later, but just like this is an overview. And now they have 2 functions. They can either repress or activate. Okay, so if they activate, they allow the RNA, the polymerase, to bind and then to transcribe the gene, or they can also prevent the RNA polymerase from binding, and so it cannot transcribe the gene. Okay, so at the end of the day, it just regulated that. So let's say here we had a gene that coded for hair color. Okay, so in this case, this protein regulated whether this hair color gene is going to be expressed or not, but the protein itself does not have a function that like it's structural. Okay, so an example for this repressor is gene is lac repressor and del repressor. Same thing again. Don't confuse the regulatory gene with the protein itself. Okay, it's not transcription factor. Is not the gene. It's just the protein that comes from the gene. Okay, so those are the 2 types of genes that we have. Something that you should know, it's not directly written, but you should know because it comes up in questions, is that structural genes have way more consequences, because if any mutations happened in the structural genes, there will be a protein directly affected. Okay, so this will cause more chance of death to the person and more chance of this gene being inherited because the person likely dies. Okay, but in the regulatory genes, it's less like dramatic. Like a person can have a mutation and it may not even be visible, or it could be like affected on the long term. So it is more common to be seen because this person lives, so we can see this gene. Okay, next thing is 2 types of enzymes you should know. There is something called a repressible enzyme and something called an inducible enzyme. We're gonna like take the example of the lac operon so you understand those, because I feel like it'll be useless to make you understand it without the example, and then we're gonna come back to this. So now we're gonna take the regulation in 2 different organisms, in the prokaryotes and in the eukaryotes. Okay, so in the prokaryotes, the example we have is the lac operon. Okay, please understand this because there's there's other operons in the bacteria, but like, so one time they got the trip operon, and you didn't know this from the curriculum, but you just had to know it from understanding the lac operon. So just know that the bacteria has multiple operons like this for like genes and stuff. So let's try to understand what happens here. Now here we have 3 different genes. Okay, we have the lac Z gene, the lac Y gene, and the lac A gene. You should know them in order. Okay, and you should know that before them there is the promoter for the structural gene and the operator. Okay, so a little thing we should know about bacteria is bacteria prefer glucose for respiration. Okay, because it's easier, no need to break it down. So as long as there is glucose in the medium, we do not use lactose, obviously. So we do not need any like proteins that use up lactose. We do not need the enzyme that breaks down lactose, or the protein that like makes the lactose enter the cell. We do not need those. So as long as there is no lactose in the environment, this lac operon is repressed. Okay, it is not active. But the moment there is no more glucose and there is lactose in the environment, the bacteria is forced to use the lactose to respire. Okay, so in this case, it will activate this lac operon. So this is the whole point of the lac operon, just so you know. So let's picture here two scenarios. Scenario number one, there is no lactose in the environment. Okay, so normally the lac repressor has two binding sites. There is binding site up here for lactose, and there's a binding site at the bottom to bind on the operator. So as long as there is no lactose, this lac repressor will bind on the operator. Okay, so now the RNA polymerase cannot bind on the operator or on the promoter to like transcribe those genes. Okay, so this is in case there is no lactose. So let's imagine that there is lactose right now in the environment. Okay, so in this case, the lactose binds on the allosteric side. Like, it does not bind on the same binding side as the operator. No, it binds on the other side here. Okay, so now it kind of changes the shape of the main active site. So the lac repressor is no longer complementary to the operator, so it leaves the binding site. This leaves the RNA polymerase free to bind on the promoter. So do notice something. The RNA polymerase binds on the promoter. This red thing is the promoter, while the lac repressor binds on the operator. It's just that they're very close to each other, so the RNA polymerase cannot bind on the promoter as long as the operator is here. Okay, so now the RNA polymerase will start transcribing. It will transcribe lac Z and lac Y and lac A. So now we have a messenger RNA, okay, and we can start coding for this. So what do those three genes code for anyways? You should also know this information. The lac Z codes for beta galactosidase, okay, while Y codes for lactose permease, and then lac A codes for acetylase. Okay, so the beta galactosidase basically breaks down the lactose, while the lactose permease allows the lactose to be able to enter the bacterial cell, so it codes for channels. And the transacetylase, like, there's not really a lot of details about this. Okay, so now the bacteria can use the lactose for respiration. So the function of this is to prevent, so it can ask you what is the function of the lac operon. So firstly, it prevents the bacteria from synthesizing and transcribing, transcribing unnecessary messenger RNA and then synthesizing unnecessary proteins, okay? So wasting amino acid and energy on enzymes and channels that we will not use. So after we took the control in prokaryotes, now we move to eukaryotes. So eukaryotes have something called transcription factors. Like, here we did not talk about transcription factors, here we talked about operators, okay? Eukaryotes do not have operators, we have transcription factors, okay? So the thing about transcription factors is they bind on promoters, okay? And they have two functions. They're either activators, so they activate the gene, or they are repressors, so they repress the gene. Depends on the function. So they bind on the promoter, which is on the DNA, and then multiple ones bind, forming a kind of complex. Okay, this complex allows the RNA polymerase to bind to them. Okay, and now it's able to move on the gene and transcribe it. Okay, so this is how the gene gets switched on. Okay, to get switched off, obviously it then prevents the RNA polymerase from moving, so now it's switched off. Okay, so what is the role of transcription factors in gene expression? So they have multiple roles. Firstly, you're going to describe how they work by saying that they bind on the promoter to either activate or deactivate the gene. Okay, they also allow the genes to get activated in proper sequence. Okay, so let's imagine this usually happens in fetus development, because when the fetus is developing, it has to be kind of developing in a specific order. I can't really tell you details, but you can't develop fingers before developing the hands. Okay, so in this case, the genes for developing the hands have to come before the genes of developing fingers. Okay, so this is what it means by proper sequence. And then number 3, it allows response to environment. Okay, so sometimes the transcription factors are affected by some sort of environmental stimuli. So let's imagine you sitting in the sun. Okay, you get tanner. Why? Because transcription factors activate the gene that codes for melanin. So now you'll have more melanin response. Okay, so this happened because of the environmental stimuli, which was the UV waves. Okay. Last thing was that the transcription factors, sometimes they're a product of like tumor suppressor genes. Okay, so tumor suppressor genes sometimes code for transcription factors that suppress tumors by activating apoptosis or preventing the gene, the tumor from growing further. So this is what it means by tumor suppressor genes. What about plants? So in plants, what controls the genes is firstly gibberellin. Okay, as I said, I explained this part in more details in the coordination video, so please go watch this, but I'll just kind of do a quick overview here. So the gibberellin is responsible. So if the question asks you how does gibberellin activate genes, you're going to say that there is receptors on the cell surface membrane for gibberellin. So the gibberellin binds on it. It does not enter. This is something very important. The gibberellin does not enter the cell, but it activates a kind of enzyme cascade that sends off enzymes into the nucleus to destroy the DELA protein. So who is the DELA protein? The DELA protein is a repressor protein that binds on PIF, which is a transcription factor. So as long as this DELA protein is bound to the PIF cannot do its function. So once those enzymes are activated and this DELA protein is destroyed, the PIF transcription factor is now free to go and bind on the promoter. So once it binds on the promoter, it can now allow the RNA polymerase to bind, and it can now activate the genes. Okay, so now this plant can activate the genes that code for hydrolytic enzymes. Okay, so the seed can germinate. Starting the part 2 of this video, which is the variation part. As I said, so you're not confused, this is part of chapter 17. Okay, this is before natural selection. So when I was explaining natural selection, I'm so sorry that this video is in Arabic, so I know my English viewers cannot watch it, but when I was explaining natural selection, I was saying that the first part before natural selection is something called variation. There has to be variation, so there is a change in alleles, so now it can choose which alleles are better. So what causes the changes in alleles are basically meiosis, mutation, and random fertilization. Okay, in this case, in this part, we're going to dive in phenotypic variation and how is the phenotype affected. Okay, so as I was saying, there are firstly genetic factors that affect the phenotype, which are the main ones, and there is also environmental part of factors which like modify the genetic parts. Okay, so the genetic parts are basically meiosis, mutation, and random fertilization. Okay, so in meiosis, basically we have the independent assortment and crossing over and random fusion. All of those things cause variation. Okay, and we have mutations, which is this, like all of those examples here in chapter 16. So in any question where it was like mention mutations that cause some sort of genetic variation, you're going to mention all those 4 diseases because those are purely genetic. There's no environmental factor affecting those. Okay. And we also have the random fertilization or the random mating. Okay. The next thing is environmental. Okay, there's also a part of the environment that can affect the phenotype. So for example, something like skin color is affected by the sun, or something like body mass is affected by how much food you have. So there is some things that are still affected by the environment, which are mainly seen in the continuous variation. Okay, so now we have the factors that affect the phenotypic variation. We have 2 types of variation. We have the discontinuous variation and the continuous variation. You're a bit familiar with this from the O-levels, okay? So this discontinuous variation gives us a type of variation that does not continue. It is distinct. So you either have this phenotype or you have this phenotype. There is no intermediates in the middle. So an example for this is, for example, blood groups. You either have one of the 4 blood groups. You either have A, B, O, or AB. There is no in the middle. There is no O and a half or B and a half. No, it's A, O, B, or AB, okay? So if you're gonna like put this on a graph, it's gonna be a bar graph, okay? And this type of variation is not affected by the environment at all, okay? And if it like in the question asked about the genetic effect, okay, so how is the genetic basis of the discontinuous variation? We're going to say that the discontinuous variation is controlled by one or very few genes, okay? So if there is only a few genes controlling this, so any change in allele has a great effect on the phenotype. So like we were mentioning here, for example, any change of alleles here caused a huge effect on the phenotype. Now we have a whole disease, okay? So this is something major. Now we have the continuous variation. The continuous variation is something that is a bit more continuous. It has ranges. There's no distinct phenotype here. So something like, for example, the height. We have so many different heights. We have 150, 151, 151.5. There's so many intermediates in the middle, okay? And here the environment plays a role. So for example, something like the height, yes, you can have the genes for being tall, but at the same time, you have to eat enough food to grow. So if you're not eating enough proteins, those genes you have will not work or not get activated, okay? So, and we put the data on this to a bell graph, okay? So there's always a range. Okay, and then about the genetic basis of continuous variation, this is important because there was a question once that like just straight out asked what is the genetic basis of continuous variation, and a lot of students couldn't like know which part to write. It's like 4 things. Firstly, that the continuous variation is controlled by the effect of many genes. So there are many genes that code for this trait. So something like height, you don't only have one gene that affects height. No, there are multiple genes, okay? So any change in the allele will not have a drastic effect. It's only a little effect because you have other genes that code for height, okay? And it gives us something called the additive effect. So I kind of explain this to you guys. It's like, imagine that every allele we have adds onto your height like 5 centimeters, okay? So if you have 10 alleles, okay, so each allele will add on 5 centimeters, so 10 times 5 is 50, so now you are 50 centimeter because the effect of all those genes are added, okay? This is kind of to simplify it. Last thing is the environment can affect the gene expression. So like I was saying in the part about transcription factor, it's that sometimes the environment can have an effect on the gene, okay? So in this case, those are the genes that can be affected, okay? So environment affects the expression. The thing is, the environmental things are not inherited, okay? So for example, if someone has the allele of being tall but is not tall because he lives in a famine, okay, he cannot inherit famine, okay? Like if your kid has the gene and then lives somewhere else, he's gonna be tall, while genetic things are inherited, okay? So if you get the disease of like sickle cell anemia, you could inherit this to your child. Okay, so this is something also you should keep. We covered everything here. The last thing left is the t-test, okay? This is the things, like, it's taught in paper 5, but it can still come up in paper 4. Some students forget to, like, look at those things, so do look at those, like, tests, like the t-test and the chi-squared and the Pearson and Spearman, the index of biodiversity. All those things sometimes still come up here in paper 4. Also the quadrants come, so, like, do look at that one. So let's solve some questions together. First question was, there are some examples of phenotypic variations in plants and animals. He wants us to identify if it was genetic factors, environmental factors, or effect of both that caused the variation. So here we have the tomato, okay, with different yields. Some of them were grown in glass houses and some were grown outside. So here we have environmental differences. And then it says that 17 genes were associated with the yield. So this is all the genetic factors. So this is a combined effect. Okay. Now here it tells us that new strawberry plants from the variety called Sweet Anne are made by asexual reproduction. The new plants grow different sizes and produce different number of fruits. Okay, so those were grown by asexual reproduction, no effect of meiosis or genetic changes or so this is important here to know. And they just simply grew to different sizes, so that means that it was environmental, because asexual means that they're genetically identical, so the effect here was purely environmental. Then here it says the domestic cat has a blood group of AB or AB. The blood type are determined by the antigen presence on the surface membrane of the RBC. That's obviously genetic because blood group is genetic. Here is like over 50 genes have varieties that are associated with excessive weight gain. Other risk factors are diet and exercise. So that's basically genetic and environmental effect. The last one was resting heart rate in humans varies between different individuals. Some factors are biological, sex, family history of heart rate. Family history means genetic. Okay, number of cigarettes smoked and medication. So those are other things are environmental. So it's also a combined effect. So the trick here was just simply to identify what was outside and what was genetic. Name a spontaneous random event occurring in cells that can be a source of phenotypic variation. That is obviously mutation. Then here, other than the event named here, describe features of sexual reproduction that contribute to the production of genetically different offsprings. Okay, so as we said in the explanation, what happens is crossing over, independent assortment, random fusion of gametes, and random mating. All those things cause the sexual reproduction to be have variable offsprings. That's why you and your siblings are not identical. So whenever there's like a lot of things written, you have to kind of simplify it for ourselves. So this question was telling us that the tigerfish is a very popular aquarium fish that has the appearance of a normal wild fish. Okay, so this fish looks like the normal wild fish. And those bullet points is telling us that the tiger barb shows a wild type phenotypes or gold with black stripes, while tiger barbs that show an albino phenotypes are gold with white stripes. Okay, so there's different colors of stripes here. Black is the wild, and the white is the albino fish. Okay, in 2012, a fish breeder discovered a tiger barb with a new transparent phenotype. This fish had a transparent body and black stripes. So this is the new kind of like phenotype. The fish breeder crossed the tiger barb, showing the new transparent phenotype, with a tiger barb that was albino. So he crossed those two together. Let's see if there's any relevance to this in the question. So after the crossing, this is the most important line. All of the F1 offsprings were wild types. Okay, this means that the wild was dominant. Okay, so when crossed with the recessive, all were dominant, or like showed dominant like features. Then when we crossed the F1 offsprings with each other, it gave us this table. Okay, so this means that we crossed two heteros together. Okay, so the fish breeder concluded that the new transparent phenotype had occurred because of mutation. Explain how the results in the table conclude this. So usually mutated alleles show recessive traits. Okay, and also he wanted to conclude that whatever happened here is like genetic. So like from the mutation we know two things. Firstly, that it's recessive. Secondly, that it's a genetic trait. It's not environmental. So firstly, because like the transparent phenotype only appears in F2 generation and F1 has no transparent, that means it's recessive. So it basically was inherited down from the parents to the offspring. That means it's genetic and it's not like environmental that caused the transparent thing. Here he wants us to do the ratio. So this is a ratio of approximately, like if you divided it, it'll be approximately 9:3 to 3:1. Okay. Explain what the results in the table show about the genes and the alleles that determine the wild type in albino and the two different transparent phenotypes in tiger barbs. So we need to know like details about the genes. Okay. So we don't really have that many details, only that there are firstly two genes, because we have the color of the body itself and then the color of the stripes, so that this phenotype is controlled by two genes. Okay, and there is not necessarily any linkage to it because the ratio is 9:3 to 3:1, so the ratio is not different. And since he did not mention anything about the gender, so it is probably not sex linked. Okay, so this is if we're talked about the genes. Then we need to talk about the alleles. Okay, the like each gene has obviously two alleles. Okay, because there is no other thing. It's either black or white stripes, or it's either transparent or gold color. So there's no other alleles. And in this case, the transparent is the recessive, while the gold is the dominant, and the white stripes is the recessive, and the gold stripe, was it gold or black? Yeah, the black stripes are the dominant. Okay, so the wild type is dominant for both genes. This is what we needed to know about the genes. Okay, this is a very easy question, but I can see a lot of students like struggling in this because they just didn't know what to write. Okay, so you simply just think about like each part, like what is the gene, what is the allele for each wild albino. Okay. So the tigers from F1 all look the same, but the F2 offspring showed variation. Okay, and this variation occurred during meiosis in the F1. Okay, this allowed the variation. So what happened? So during meiosis, variation occurs because of first thing, independent assortment, and the crossing over. Here, the crossing over does not have that much relevance because they are on different chromosomes, because they're not like autosomally linked. So the main thing that will cause the variation here is the random assortment. Okay, so let's picture this here. We have homologous chromosomes. Homologous chromosomes means that chromosomes that have the same genes, okay, one of them is from the mother and one of them is from the father. Okay, so obviously before meiosis, a copy of each occurs. Okay, so there is a copy, and we have one from the mother and one from the father. Okay, so here we have two chromosomes. This one has the gene for the body color, and this one has the gene for the stripes. So it's important to know at which part this thing happens. So at prophase, the pairing up occurs, okay? While at metaphase, the independent assortment occurs, okay? So during metaphase, at the equator, those chromosomes randomly assort, okay? So let's kind of picture this here. Those chromosomes move here, and those move here. So they randomly assort. Okay, so now we have a combination of different things. So this like striped black stripe can go with the white, the transparent gene, and the white stripes can go with the or gold like gene. So we can have different mixes that weren't originally in the parents. So once now those during anaphase, the homologous chromosomes separate, they can have different combinations. Okay, so this is how like variation occurs because of independent assortment. Of course, here adding crossing over will not make you lose homework. You can still add it, so it's kind of a memorized answer. Question is also a hated question. It's telling us that the interpupillary distance is the distance in millimeter between the centers of the pupil and the eyes. Okay, and here's telling us this. And it's showing us that the interpupillary distance in different people, okay? And you can see it clearly on a histogram. So whenever there is data on a histogram or a bell graph, this means that it is a continuous variation, okay? Because you can clearly see a range. So name the type of variation. So as I said, it is a continuous variation. So it doesn't explain how genes and the environment contribute to this variation. So this is what we said a while ago, that don't like in those answers, please always like underline so we don't forget anything. So genes, we have to answer this, and then we have to answer the environment. Okay, so firstly, genes. We have many genes affect this, and they have a additive effect, and changing one gene will not have a major effect on the phenotype. So now we answered the gene part. What about the environment part? The environment does have a significant effect on the phenotype, and the environment can affect the gene expression, can modify the gene expression. And you can always mention examples, of course. And B is telling us the individuals with an IPD of 70 millimeters or more have a mutation in the PAX gene that results in less PAX protein being made. So less PAX protein being made increases this distance. The normal role of the PX protein is to increase the expression of many other genes involved in embryonic development. These genes affect a range of phenotype features such as facial structure and hearing and eye color. Okay, it's coming in a story that's confusing now. Say the term that is used to describe a gene, such as the PAX3 gene that controls the expression of other genes. So what is the term used to describe those genes? Even if you didn't understand the story, he already said that they control. So you can clearly tell that this is a regulatory protein, a gene. Okay? And the and is in bold, so do not forget to complete your answer. Suggest how this protein controls the expression of the other gene. So you can simply like solve this without understanding the prior part. So it is a regulatory gene, and it codes for a transcription factor. This transcription factor can either be a activator or a oppressor. In this case, it's a activator, which binds to the DNA at the promoter region. Okay, so allows the RNA polymerase to bind and stimulate and like transcribe. This question describes how microarray analysis could be used to identify genes switched on. Okay, I'm not going to get into details about this. I already have a video on gene tech, but always remember when it's telling us genes switched on to make sure to use the mRNA method, not the gene cutting method. Okay, so in the microarray, we have two different ways. We can either cut the gene from the cell, from any cell, and then just test the presence or the absence of the gene, or we can go to a cell that expresses this gene. Okay, and we can get messenger RNA, and from this messenger RNA make a complementary DNA. Okay, and then use this complementary DNA in the microarray. And then we're going to complete in the microarray chips, add the DNA with the probes, hybridize, and then wash off and see the fluorescence. And the fluorescence will indicate the gene expression. The more the fluorescence, the more the gene expression. The DNA 95%, 98.5% similar to humans, including the possession of the PAX gene. So the chimpanzees also have this gene, but the chimpanzees show a very high expression of this gene. Okay? Suggest how knowledge of this gene helps scientists explain how humans and chimpanzees are very different, have different facial features, and even though they are very similar in DNA. Okay, so here is something important. We know that regulatory proteins do not have that major effect because they can, like, there is, like, they only control, they do not contribute to a direct protein, like a structural one. But in this case, he did say that the role of this protein is to increase the expression of many other genes involved in embryonic development. Okay, and those genes control facial structure and hearing and eye color. Okay, so there are like a lot of genes that control many major aspects of our looks. Okay, so if we have a change in one of those genes that control the expression of all of those other things, we're going to have a big phenotypic effect. Okay, so this is going to be our answer, because a small change in one of the PX gene will have a major phenotypic effect because it regulates many other genes, and those genes have an effect of the face and hearing and eye color. Okay. So those chimpanzees may have the same gene, but they can have a higher expression because of something. That's why we look different from them. Okay. This question was telling us that we have dwarf varieties. Okay, so the scientists treated 3 varieties. So we have 3 varieties of dwarf, and they treated them with gibberellin. Okay, we have the wild type. This is the control, and there was the dwarf variety 1, A and B. So those were 2 dwarf varieties, and this is a wild type for comparison. With reference to the figure 6.2, suggest and explain for the different response to gibberellin shown by dwarf A and B. In a question like this, I have to think about what do we know about gibberellin, or what do we know about dwarf varieties. So what we know about gibberellin is that it activates the expression of genes, and what we know about dwarf varieties is they have a functionless gibberellin because they have a mutant recessive allele. Okay, so those plants do not have normal gibberellin. So if you think about it, they only have functionless gibberellin. So if we added functional gibberellin, it should normally increase the size. Okay, so in this case, the wild type has normal gibberellin. So when we added the gibberellin, it made it increase more in height. Same thing with this variety. When we added the gibberellin, it increases the size, because now you're going to explain how the gibberellin affects the size by binding to the receptor, breaking down the DELA protein. So now the PIF transcription factor can bind to the gene and increase internodal length and elongation and stuff. So the dwarf variety A has the LE small, which is the recessive. So we cannot produce active gibberellin, but it still can work normal gibberellin. But here was weird because the dwarf variety, even after adding the gibberellin, it did not work. So that means there was probably a problem in the receptor itself. So even after adding the gibberellin, they couldn't bind on the receptor, so it couldn't even make a response. Okay, so this was the different response to gibberellin here and here. Now this question was, using hemophilia as an example, explain the relationship between a gene, a protein, and a phenotype. This was, like I explained to you guys, you're just going to mention each one of them. You should memorize those questions because they come like that sometimes. So a gene, you mentioned that a gene codes for a protein, which determines the phenotype. So you're going to mention each thing here. What is the gene? In this case, it's the F8 gene. Okay, it's present on a X chromosome because it's sex-linked, so it's more common in males. Okay, it is a recessive allele that codes for a non-functioning factor 8. So those people bleed a lot because they do not have enough clotting factors. And this question is telling us that the pea plant, the genotype has LE small. Explain how the LE genotype and its gene product results in dwarf plants. This was what I just said, that those plants have the recessive alleles, so it codes for a non-functioning enzyme. So sometimes when we explain, we just say that they do not have active gibberellin, But like to be more specific, they do not have the enzyme that activates gibberellin. So they have inactive gibberellin. Okay, so that's a little bit of to be more specific. And then you complete the answer like we did. And this was the part about the DELA protein, already explained this. Okay, this is a good question. So when I was explaining the lac operon, I told you guys that there are other operons in the bacteria. Okay, so here is telling us that there is a operon called the trip operon. Okay, and here we have the structure of it. So we're going to look at it. Okay, there is an inactive repressor, there's an RNA polymerase, there is the tryptophan. Okay, we are confused. Let's complete and see what he wants. Describe the differences in structure and control between the lac operon and the trip operon. Okay, whenever he mentions differences, please add both sides. Please add both sides. Like look here, if you do not write the other side, you're not going to get any mark because the and is in bold. Okay, so here he gave us the trip operon because we did not study this, and he needs us to compare this to the lac operon. So we need to be strategic about this. Let's talk about the structure first and see how this will affect the control. So he wants only differences. This is showing us a gene, a regulatory gene that codes for an inactive repressor. Okay, so the repressor is normally inactive unless it is activated by the tryptophan. But this is like an opposite to the lac operon, where the lac repressor is usually active. Like the lac repressor is usually bound already on the operator. We can get the lac operon in comparison, so we can kind of easily compare. So here we have the lac operon versus the trip operon. So here, as we said, it's normally here active, like it's already bound to the operator. While here, when there is no lactose, okay. While here, if there's no inducer, it's like inactive. So this is the first thing. There is something called the attenuator, which was not present in the lac operon. There is like 1, 2, 3, 4, 5 structural genes, while on the lac operon there are only 3. Okay, so that's something. Last thing in the structure is that the lac operon, we used a inducer, which is the lactose, because when lactose is there, it binds and it activates the lac operon. While here, when the tryptophan is present, it actually represses it because it binds to the repressor and activates the repressor. So now this is not activated. Those genes are actually inactivated. Okay, so this is all about the structure. What about the function? In the trip operon, the trip operon is normally active. Like when there is no tryptophan, it's normally active. And when the repressor binds on, it makes the RNA polymerase not able to bind to the promoter. So now those genes are not transcribed. While in the lac operon, that's the opposite. The lac operon is normally repressed because as long as there's no lactose, the repressor is bound. So the RNA polymerase cannot bind. But when there is lactose, now the RNA polymerase can bind. Okay, so the lactose normally causes the genes to get expressed, while the tryptophan normally causes the genes not to be expressed. Okay. Now, if you completed, this is an easy question. Suggest why structural genes in the operon are transcribed together. So it's something you should know is whenever we activate this lac, all those 3 get activated together. They all get transcribed and synthesized. It's because we have one promoter. So when the RNA polymerase passes, it can only pass all of those in the same direction. Okay. Trip A is an example of a structural gene, and Trip R is an example of a regulatory gene. He wants the differences. Okay, just mention the definition here. He's telling us that Trip A codes for enzyme tryptophan synthase. Tryptophan synthase catalyzes the formation of amino acid tryptophan. Okay, so this enzyme forms the amino acid. Explain why tryptophan synthase is an example of a repressible enzyme. So firstly, whenever there is a lot of tryptophan, it makes an end product inhibition to inhibit the enzyme. So this means this enzyme is not always working. It is repressible. It can be repressed. Okay, so the tryptophan binds to the repressor and activates it, which means that this enzyme is not always active. Okay, so the tryptophan prevents this gene from being synthesized. So, like I said, I'll do a quick recap on meiosis. So it's like the video is full of chapter 16 and also one crossing video. If you're an Arabic watcher, you can watch my video on inheritance. I did way more crossing there, so I'll just throw like one here. So for meiosis, the whole point of meiosis is reduction division. It's for sexual reproduction. So it splits up the chromosomes into half. So once fertilizations occur, they can never duplicate. So that's the first thing. The second thing about meiosis that people forget is the variation. Like variation is like meiosis leads to variation, so like switches up the alleles, so can favor natural selection. The thing about meiosis is it only reshuffles existing alleles. It does not create new alleles, unlike mutation, for example, where new alleles are formed. But here they're simply just existing ones, but they kind of mix up, you know. So that's something. Meiosis occurs in the gonads, it so happens in the ovaries and in the testes. For meiosis, it's similar to mitosis in like having interphase before it, but different because it has two meiosis 1 and meiosis 2. So just before meiosis 1, the cell passes in the interphase. Interphase is not part of meiosis, just occurs before. Okay, and there's also an interphase before meiosis 2, but it's way shorter because there's no need for like duplication and stuff. The duplication happens in meiosis 1. Before meiosis 1, each chromosome has just one chromatid, so those are called homologous chromosomes. Okay, homologous chromosomes are like two chromosomes that have the same genes. Each one is from the father and one is from the mother. Okay, so just before meiosis, they copy. So now each chromosome is made up of two chromatids. Okay, so that's the first thing. And also the centrosomes like duplicate just before prophase 2. So now once we entered the prophase, it's the same as in mitosis, prophase, metaphase, anaphase, and then telophase, but all of those are one. Okay, so like in general, like meiosis 1 is longer because that's where more things happen. Meiosis 2 is a bit like shorter. So during meiosis 1, firstly, the homologous chromosomes, they pair up. So now each chromosome is going to be with its like homologous pair. Okay, so now they're called bivalent. So this is also called synapsis, the whole pairing up. This happens in the early prophase, and then in late prophase, the something called crossing over occurs. Okay, so if we look here, this was from the father and this was from the mother. Okay, so now part of the end of the chromosome will kind of switch over. So now each chromatid has a part of the other, and the point of like fusion between them, so this point that I'm pointing at with the orange dot, is called the chiasmata. Okay, this is the point on where they exchanged the genetic information. So this happens between non-sister chromatids because those two are called sister chromatids, but this happens between non-sister chromatids, between this and this. Okay, so this of course, both of like this causes like variation because now we're switching off like genes, and this mostly is effective if the genes were on the same chromosomes. So now we're switching over chromosomes, like genes from same chromosome. Now we have metaphase, like nothing really special. They line up at the equator, but the homologous pairs do. And here something important happens called the independent assortment, where the homologous pairs pair up at the equator in a random manner. Okay, this is the second cause of variation. And then third thing is the separation. Okay, here the sister chromatids are not separated. Here the homologous chromosomes are separated. So take care here, because here we say that the homologous chromosomes get separated. So now each homologous pair is going to go separate ways. And here the like now we're starting the telophase, and like everything happens like mitosis, nuclear envelope reforms, nucleolus, and cytokinesis. So this is telophase 1. Now we go into meiosis 2, and here same thing happens like meiosis 1, but it's just shorter. But here everything happens. Now everything happens between sister chromatids. So at prophase 1, same thing happens, and then at metaphase 2, the spindle fibers extend to attach to the centromere, and then the attached the chromosomes at a perpendicular angle from the first ones. Okay, so that's something special. And then telophase 2 happened, and then cytokinesis occurs, where now we have 4 genetically non-identical or like genetically different haploid cells, while in telophase 1 we only had 2. So in this question, this very popular that comes and asks how meiosis and fertilization can result in genetic variation, we're going to just list out all the things that cause variation. So like I said, the chiasmata between non-sister chromatids, okay, of homologous chromosomes in prophase, that's the first thing that causes variation. Secondly is the exchange of genetic material, a linkage group that causes the new combinations of allele. This is the result of this. Okay, because now we have different combination. Second thing is the random assortments of homologous chromosomes at the equator in metaphase 1. Some random assortment still occurs at metaphase 2, and then mutation can happen, and this is the first part. The second part was how fertilization can result in genetic variation, and here it's just the randomness of it. So random mating and random fusion, the more random it is, the better. So this question was telling us that there is a rosy periwinkle flower that is controlled by 3 genes. Okay, the presence of the R allele results in the red pigment in the center. So the R dominant results in the red pigment in the center, or the red eye. Okay, so having the probably recessive one means no red eye. Now we have the D and P. They're only expressed when the R dominant is present. So if we have R small, none of those are going to be present, and the flower is going to be white. Okay, so it has no red eye and white. What about if the R is present? When the D and the D dominant and R dominant are present, the flower is dark pink petals with red eye. But when the P and R are present, the flower has pale pink. When the D, P, and R are all present, the flower has a dark pink. So now they're kind of all co-dominant because we have different, they all interact with each other differently. Okay, so now we're just going to look here and kind of compare. So it has the R. That means it's going to have a color, okay, and it's going to have a red eye. So it has the R dominant and P, so we're with this example. So it's pale pink with red eye. Here it also has the R, it has the 3 of them. So it's going to be red eye again with dark pink petals. Now it has R recessive. That means it will have white petals and no red eye. Last thing, we have the R dominant and only this, no D or P. Okay, that means it will have white petals but will have red eye because of the last one. Okay, so always kind of write them down next to the question so you kind of know. This was just a mutation question. Okay, that's it. I hope this video was helpful.